Hi, my name is Andréanne Proulx. I'm a master's student at the University of Montreal in the CMXP and Jacquemont labs, and today I will present my work on the alterations of the principal macroscale functional gradient in carriers of neuropsychiatric CNVs. Deletions at the 16p11.2 and 22q11.2 loci are copy number variants that result in large missing segments of DNA. They're located on the 16th and the 22nd chromosome, respectively, and imply the absence of all the genes in the concerned deleted areas. This amounts to 29 genes in the case of 16p and 50 genes in the case of the 22q deletion. While these mutations increase risk for physical abnormalities, there are also some of the largest known genetic risks for an overlapping set of developmental neuropsychiatric conditions, such as autism, intellectual disability, and schizophrenia. It remains unclear why 16p and 22q deletions located at different genomic sites increase the risk for some of the same developmental conditions. One hypothesis is that they may converge on shared mechanisms at some level, from the genes to the large-scale brain networks to the phenome. Prior work has focused on functional connectivity, a measure that quantifies the codependency in blood oxygenation levels between pairs of regions, serving as proxy for communication levels between regions in the brain. These findings have indicated that there is some specificity as well as shared dimensions between these two deletions, involving mainly the default mode network, the ventral attentional, the frontoparietal, and the somatomotor network. Recent studies have highlighted that many properties of the brain, including functional connectivity, is organized in a dominant orderly manner across the cortex, from the lower order unimodal cortices with sensory and motor functions to higher order transmodal association cortices. Interestingly, this cortical spatial trend aligns with the spatiotemporal patterning of cortical maturation that leads to enhanced connectivity differences between lower order and higher order functional systems. In this study, we hypothesized that the principal connectivity gradient is the key brain dimension between 16p11.2, 22q11.2 deletions and the developmental neuropsychiatric phenome. Our first aim consisted of dissecting these high-risk deletion effects on the principal connectivity gradient, and the second aim consisted of contextualizing these differences by exploring phenome-wide associations in larger and richer phenotypic population, the UK Biobank. We pulled resting state fMRI data from two genetic cohorts, the Simons VIP Consortium and the UCLA site, which comprised 19 carriers of 16p deletion and 42 carriers of 22q deletion. We also had controls from each of the scanning sites. We pulled over 38,000 subjects from the UK Biobank population in order to achieve our second aim. All this resting state fMRI data underwent standard preprocessing steps using the neuroimaging analysis kit pipeline. We then extracted the principal connectivity gradient. First, what we did is we parcellated this data with the Schaffer atlas at the resolution of 400. Then we computed the correlation between these time series. And ultimately, this gave us the individual connectomes. And finally, we applied the diffusion map embedding to the affinity matrices of these connectomes using the BrainSpace toolbox. Each region was given a gradient value indicating its rank along this sensory motor to association axis. We adjusted gradient values for the effects of age, sex, motion, global signal, and site. Now, for the first analysis, we did a case control regional statistical comparison of the principal gradient values, and we did that by computing the cohency statistics. And then we correlated our statistical maps between the two deletions and assessed significance with spin permutation testing. That relative to controls, Both deletions exhibited alterations of the first connectivity gradient. The largest effects were associated with deletions of the 16p11.2 loci, which is preferentially associated with autism spectrum disorder. The functionality of network-based shifts described predominantly higher gradient values in sensory motor and dorsal attentional networks, which contrasted with lower values in limbic frontoparietal control and the default mode network. Results were robust considering different sets of confounding factors. Finally, case control maps of principal connectivity gradient alterations converged. They were significantly correlated with a correlation strength of 0.36. Now, for the second aim of the study, the goal consisted of exploring phenotypic associations of genetic profiles within the larger population. A common challenge studying rare mutations are low sample size and the lack of available phenotypic information. Emerging populational datasets such as the UK Biobank that have rich phenotypic profiling present an opportunity to characterize brain phenotypes at an unprecedented scale. We tailored an analytical strategy to carry over the deletion profiles to the UK Biobank population. We quantified subject-level similarity with each of the 16p and 22q deletion profiles extracted at AIM-1, and then performed a phenome-wide-association study with 257 variables spanning 10 categories of phenotypes encompassing physical, lifestyle, cognitive, and mental health domains. Now, as for the results, so here you have the Manhattan plot that is showing 257 variables and their associations with the brain gradient deletion expression levels. So what we found is that deletion expression presents phenome-wide associations for both loci, meaning that they have significant correlations across multiple categories. More precisely, we found 66 associations with 22q11.2 deletion and 57 with 16p11.2 deletion. A majority of these associations were in the physical general bone density and size and lifestyle general categories. Furthermore, we obtained an increased number of hits compared to the null distribution from randomly generated case control maps obtained by permuting group labels. Finally, we looked at the similarity between the different phenotypic profiles and found that they presented similar patterns of association across categories. They showed increased overall correlation strength compared to the null case control maps. In summary, we found alterations of the principal connectivity gradient with spatial patterning of effect sizes, describing a contracted principal gradient. These results are consistent with studies that have investigated the principal connectivity gradient in neurodevelopmental conditions such as autism and schizophrenia. Furthermore, we found that gradient profiles pertained to phenome-wide associations in the UK Biobank population, and that deletion-specific gradient changes led to increased phenotypic associations compared to null-generated profiles. Finally, we found that these phenotypic patterns of associations were similar between the distinct deletions. Our result indicated the relevance of lower dimensions representations for brain connectivity in understanding genome to brain to phenome. A future direction could be to extend this work to other CNVs with high, medium neurodevelopmental risk to see if they converge on similar dimensions.